All right, guys, we need to begin. So today we're going to talk about the muscular tissue, And we know that in our body we have four different types of tissues. The first one was the epithelial tissue, followed by the connective tissue, and then we have our muscular tissue, and our next lecture is going to be about the nervous tissue. So right off the bat, when we talk about the muscular tissue, what are we talking about? We're talking about a tissue that is excitable. The same thing with the nervous tissue. The nervous tissue and the muscular tissue are both excitable. What does excitable mean? It means that they respond to membrane voltage change. They respond to electrical stimulation. They respond to membrane voltage change. They respond to electrical stimulation. And, this is the concept of the action potential. So when action potential is propagating through the membrane of a muscle cell or a nerve cell, the ion channels would open, and that would cause an influx of sodium from the outside, the extracellular space, into the inside, the intercellular space, and that is going to cause a depolarization. And that would be followed by the opening of the potassium channels known as repolarization. So that is the action potential. The action potential elicits excitation in the muscular tissue and excitation in the nerve tissue. So what happens when there is excitation of the muscle tissue? The muscle tissue would contract. But when there is excitation of the nerve tissue, what happens? The nerve tissue does not contract. So the nerve tissue would mediate a signal. So all the muscle cells have the following characteristics. Number one, the muscle cells are excitable. Excitable means they respond, they are responsive. They are responsive to chemical signals, stretch, and electrical changes across the plasma membrane. So when we change the membrane voltage across the plasma membrane of our muscle cells, we're gonna elicit excitability. And this muscle cell, upon getting excited, it's going to be responsive. It's going to respond to this chemical signal, the stretch, the heat, thermoreceptor. Remember the thermoreceptor, the chemoreceptor, the osmoreceptor, the nociceptor. What are they going to do when they get activated? These receptors are going to cause the opening. They are going to cause the opening of the ion channels, allowing for an influx that's going to ultimately cause a depolarization. So the first property of muscle cells is excitability. What is excitability? Yeah, the action potential passing through the nerve or the muscle membrane, causing different influx and efflux of ions across the membrane. Then what is conductivity? Conductivity is when local electrical excitation set off a wave of excitation that travels along the muscle fiber. So that means the muscle fiber, how many different types of muscle tissue do we have? We have skeletal muscle tissue. That's the skeletal tissue, the muscles that are attached to your bone, muscles that are attached to your bone. And then you're gonna have cardiac tissue, and that is muscle of your heart. And then there is smooth muscle, and that is muscle that is all around, the blood vessel. So the electricity is going to flow through the muscle fibers of the heart. So that shows you that the heart is a synthetium. What is a synthetium. A synthetium is when the cells are working together in unison, propagating this action potential. So when your heart is going to contract, what happens? You have electrical signals that are at the base of the heart with the sinoatrial nodes, and the sinoatrial nodes are going to elicit action potential, and the action potential is going to propagate through the cells of your heart, and then they're going to pass from the atria to the septum to the ventricles through the Purkinje fibers. And as these electrical stimuli are passing through your heart tissue, they are going to cause your heart tissue to contract. So the atria would contract when the electrical signals depolarize the atria. Then when the atria repolarizes, it will relax. Then the ventricles are going to depolarize, and following the depolarization of the ventricles, you're going to have the relaxation. So that shows you conductivity is the ability of the skeletal muscle tissue, whether smooth muscle tissue, whether cardiac muscle tissue, whether skeletal muscle tissue, all the muscle tissue is able to propagate and pass electrical stimuli down from the tissue to the nearby tissue. That is known as conductivity. Then there is contractility. Contractility is the response of the muscle cells, to electrical stimulation. So when the muscle cells are electrically excited, they get shorter, they get smaller, and that is because the microtubules, these cytoskeletal proteins that are inside the cell, after the cell is depolarized and excited, these muscle proteins are going to contract, making the sarcomere smaller, making our muscle cells smaller. Then there's the property of extensibility. Extensibility is when your muscle cell is able to stretch between contractions. We have elasticity, and that is the property when your muscle cells are able to return to their original rest length after being stretched. So what is excitability? Excitability is action potential. What is excitability? So somebody explain excitability for me, please. Guys, what is excitability? Excitability is when the action potential passes by your muscle or nerve tissue, leading to membrane voltage change. Action potential cannot propagate through connective tissue. It cannot propagate through epithelial tissue, only muscular. And what is the other form of tissue that gets excitable besides muscular? Yeah, the nervous tissue. Then we have conductivity, and that is when the muscle is made up of cells, and all the cells propagate. They pass the action potential from themselves to the nearby tissue, and that's how you're going to have the conductivity. You're going to have the electrical action potential passing from one region of the heart to the next, because these cells are able to pass by this action potential. Then you have contractility. Contractility is the shortening. Contractility is the shortening of the muscle cell when the muscle cell gets stimulated. And then there's extensibility. Extensibility is when the cell is capable of being stretched between the contractions. And so our skeletal muscle, our cardiac muscles, our smooth muscle, after they are contracting, is like a cycle. They contract and relax. They contract and relax. They do not remain contracted eternally. So they have the property of contracting followed by relaxation, except in the dead people in rigor mortis. In rigor mortis, you only have contraction, there is no relaxation because these cells are dead and we need ATP. We need ATP in order to relax this muscular tissue. Elasticity is the ability of the muscle cell to return to its original rest length after being stretched. So we have contraction, then we have relaxation or the stretching, followed by returning to that baseline level. And we will see more about this. So when we examine histologically the skeletal muscle cell, we see that the skeletal muscle cell is under voluntary control. So your brain regulates your skeletal muscle activity. Unlike your cardiac muscle, so your brain does regulate your cardiac muscle, it does regulate your smooth muscle, but it is the parasympathetic nervous system. So the parasympathetic nervous system is responsible for the mediation of the activities of the involuntary portion of your nervous system. So heart cells that contract without your control. So your heart is pumping, you're not making it pump. You make your arm go up, you make your arm go down, but you cannot make your heart pump faster or pump slower because your arm is skeletal muscle. That is under your direct control. But your heart and your smooth muscle, your blood vessels, they are not under direct control. So your skeletal muscle system, so when you examine the cells of these skeletal muscle tissue, you see that they are attached to bone. So you're able to move your bones, not because the bones are excitable. The bones are connective tissue. The bones are non-excitable tissue. It is your muscles that are excitable. And then you have besides the muscles, you have smooth muscle. Smooth muscle would dilate and constrict based on parasympathetic control. So there are striations. Striations are alternating light and dark transverse bands that result from the arrangement of the internal contractile proteins. And we're going to see this in the diagram, that in order for your skeletal muscle to contract, you're going to need these cytoskeletal proteins to get shorter. And as they get shorter, they are able to make the sarcomere smaller, and that's going to lead to your skeletal tissue getting tighter and smaller. So the skeletal muscle tissue is under voluntary control because it is usually subject to conscious control to your cerebrum, your sympathetic nervous system, while involuntary control is guiding your heart muscle and your smooth muscles. This is a very important concept, guys, that you have to differentiate that your skeletal muscle tissue is a tissue that is under voluntary control, under your cerebrum control, under your sympathetic control, while your heart tissue, cardiac myocytes, and your smooth blood vessels are under involuntary control. And that is the parasympathetic nervous system. So the skeletal muscle cells are also called muscle fibers or myofibrils. So why do we have a lot of nuclei? Why do we have a lot of nuclei in our skeletal muscle tissue? What is inside the nucleus? What is inside the nucleus? What is inside the nucleus, guys? If you open the nucleus, what is inside it? What do you find inside the nucleus? What? You guys don't know what is inside the nucleus? Oh no. What is inside the nucleus? If I open my nucleus inside the cell, what is there? I'm sorry? What is inside the nucleus? The DNA. So why is the DNA useful? So I can replicate the DNA, double up the DNA for cell division. So I can make transcription of proteins. So this DNA, I need it for cell division. That means when my skeletal muscle cells are damaged, when my skeletal muscle cells are damaged, they are going to enter the cell division cycle in order for them to divide, to make up for the tissue that is damaged. So if my muscular tissue is damaged and I need to repair this damage, build up more tissue in the place of the tissue that is not there, what do I need? I need to trigger cell division cycle. So that's why your muscular tissue has a lot of nucleus. Then what else do we see? We see the striations. These striations represent my cytoskeletal proteins. These are the cytoskeletal proteins that pull the corners of your muscle fiber leading to the contraction. So once I have a depolarization, the depolarization is going to cause an increase in calcium secretion, and the calcium is going to cause the property of contraction. And that is what makes my muscle fiber able to contract. So the skeletal muscle also contains fibrous connective tissues. You have your endomysium, your perimysium, and your epimysium. The endomysium surrounds each muscle fiber, and the perimysium is made up of bundles of muscle fibers that are made into fascicles, while the epimysium surrounds multiple fascicles that is your entire muscle. So for example, I have the first level, I have my muscle fiber, muscle fiber, muscle fiber, muscle fiber, muscle fiber. These muscle fibers are surrounded by your endomysium. So each muscle fiber is surrounded by, this is the endomysium. You guys see this zigzag line? That is your endomysium. Then you have multiple. So each muscle fiber is surrounded by, the black is your endomysium. This is your endomysium. This is your endomysium. This is your endomysium, endomysium, endomysium. That's the inner layer. That's the inner fibrous layer around the muscle fiber. Then around the endomysium, you have the zigzag perimysium. Then you have multiple units like that. You have your muscle fiber, muscle fiber, muscle fiber, muscle fiber, muscle fiber, muscle fiber, surrounded by the black endomysium, 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 and the endomysium is surrounded by the zigzag perimysium, perimysium, perimysium. Then those are known as fascicles. So a fascicle is made up of perimysium that surrounds each muscle fiber encapsulated by the endomysium. Then I enclose all of them with a layer that is known as the epimysium. Epimysium. That is the most outer layer known as the epimysium. That is the epimysium. So collagen is somewhat extensible and elastic, and it stretches slightly under the tension and recoils when released. So collagen is what type of protein? Primary, secondary, tertiary, or quaternary? Collagen. Primary is a long bead, a linear chain. So it has to fold in order to become a protein. So primary is a form of a long linear chain that is known as the primary structure, and it is just a linear sequence. That is not a protein. It's got to fold upon itself first to go from primary protein to secondary protein to tertiary protein to quaternary protein. Collagen is what type of protein? No, it's not tertiary. Myoglobin is tertiary. Myoglobin is tertiary protein. What's the secondary protein? What is a secondary protein? Yeah, the beta sheet and the that is your collagen. So collagen and keratin are both secondary structure proteins. So collagen is important for tensile strength, tensile integrity. So it stretches slightly under tension and recoils when released. It resists excessive stretching and protects the muscle from injury. It returns the muscle to its resting length and contributes to the power output and the muscle efficiency. Okay, the muscle fiber. The muscle fiber has multiple components. Obviously, there is the sarcolemma. The sarcolemma is a plasma membrane of muscle fiber. You see, the sarcolemma is a plasma membrane of a muscle fiber. So the sarcoplasm represents sarcoplasm. What is the sarcoplasm? It's like the cytoplasm. Sarcoplasm is the cytoplasm. So the sarcoplasm is the cytoplasm of a muscle fiber. Then we have myofibrils. We have myofibrils, and myofibrils are long protein cords occupying most of the sarcoplasm. And then you have glycogen. Glycogen is a carbohydrate that is stored to provide energy for exercise. And you have myoglobin. Myoglobin is a red pigment that provides some oxygen needed for muscle activity. So in the sarcoplasm, there is myofibrils, and these are the bands. That's what make the striations. So you have long protein cords that are occupying most of the sarcoplasm. So if I examine under the microscope, if I look through my sarcoplasm, I will see those three. I will see the myofibrils, I will see the glycogen, and I will see the myoglobin. What is myofibril? Myofibril represents the long protein cords that are occupying most of the sarcoplasm. For glycogen, glycogen is a carbohydrate that is stored to provide energy for exercise. So we know that we aggregate our glucose to form glycogen. And where do we store glycogen? We store glycogen in the, where do we store glycogen? We store glycogen in the liver. So glycogen is like the grape stem. You know, the grape stem has multiple grapes in it. So if you want a grape, you would pull one grape out from the entire grape stem. Now, what happens with glycogen is that you pull out one glucose from the entire polymer of glycogen. So glycogen is multiple glucoses stacked together in a bundle. Where do we store our glucose? Where do we store our glucose? Yeah, we store the glucose in the liver, the glycogen in the liver, and where else? In the muscle tissue. So that's to show you that my muscle core does not just have these contractile proteins, the actin, the myosin, and the other regulatory proteins, your troponin, your tropomyosin. That's not the only thing you see inside a muscle fiber. You also see glycogen, and that is the storage form of glucose that when we anabolize, when we build up, when we store glucose, we assemble the glucose monomers, we stack them up into a polymer known as glycogen. And then we have myoglobin. Myoglobin is what kind of protein? Is it a primary? So we never have proteins in the primary. We need them to fold up. So it's either secondary, tertiary, or quaternary. What are the examples of secondary proteins? Examples of secondary proteins are collagen and keratin. And what is an example of the tertiary proteins? Myoglobin. Excellent. So myoglobin is an example of a tertiary protein. What's an example of a quaternary protein? Hemoglobin. Excellent. Because hemoglobin has how many subunits? This is quaternary. So how many subunits? 4. 2 alpha, 2 beta. And that's what makes for us hemoglobin. So myoglobin is red in color and it provides some oxygen. So when I look inside my muscle fiber, what am I going to see? I'm going to see myofibrils. I'm going to see glycogen. I'm going to see myoglobin. So myoglobin is a red pigment that provides some oxygen needed for muscle activity. There are many mitochondria. Mitochondria are bean shaped near the sarcolemma, and the deeper ones are tubular and dynamic in shape. Why do we need mitochondria? Why do we need mitochondria inside the muscle fiber? Yes, because the mitochondria is the powerhouse of the cell. The mitochondria provides ATP for the cell. So there are multiple nuclei. These multiple nuclei average about 30 to 80 per millimeter, and they serve a role in fiber repair. They serve a role in the fiber repair. Components of a muscle fiber. The components of the muscle fiber are your sarcoplasmic reticulum. The sarcoplasmic reticulum is like the endoplasmic reticulum. It is smooth endoplasmic reticulum that forms a network around each myofibril. So the sarcoplasmic reticulum is a smooth endoplasmic reticulum that forms a network around each myofibril. So what does that tell us about the endoplasmic reticulum of the muscle fiber? That shows you that the endoplasmic reticulum of the muscle fiber lacks ribosomes. We don't have rough endoplasmic reticulum, correct? We don't have rough endoplasmic reticulum. That's why they said the sarcoplasmic reticulum is smooth. And we already know there's 2 types. There's rough and there's smooth. So we have smooth endoplasmic reticulum that forms a network around each myofibril. So that means my myofibril is surrounded by smooth endoplasmic reticulum. What is the job of the smooth endoplasmic reticulum? The smooth endoplasmic reticulum is important for detoxification processes, removing metabolic toxic waste, and also the smooth endoplasmic reticulum is important for lipid biosynthesis, making fats so that we can utilize them and catabolize these fats and get ATP. And we have terminal cisterns. Terminal cisterns are dilated end sacs. These are dilated end sacs of the sarcoplasmic reticulum, which cross the muscle fiber from one side to the other. And they act as a calcium reservoir, and they release calcium through channels in order to activate the contraction. So we store our calcium inside terminal cisterns. And these terminal cisterns are dilated end sacs of the sarcoplasmic reticulum, and they cross the muscle fiber from one side to the other. They cross the muscle fiber from one side to the other. So they act as a calcium reservoir, and they release the calcium through the channels to activate the contraction. So within the cisterns, calcium is bound to a protein called calsequestrin. This name is very important, calsequestrin. So from the name, calsequestrin is calcium sequester. So it holds, like now we are sequestered in this room. The correctional officers sequester all the inmates inside the prison cell. So here we have the calcium sequestered by calsequestrin inside the terminal cisterns. Only when an action potential comes, only when the action potential arrives, that is when we let go and release of the calcium. So there are also transverse tubules, and now these are proteins. These are proteins. The proteins are tubular enfoldings of the sarcolemma, which penetrate through the cell and emerge on the other side. So each T-tubule has two terminal cisterns associated with it. So now you know when the action potential arrives to the T-tubule, is going to increase the efflux of calcium outside from the T-tubule cistern into the myofibril in order to propagate the action potential and cause the contraction of the muscle cell, the shortening of the sarcolemma, and the shortening of these microtubules. So what are myoblasts? Remember in the bone lecture when we talked about osteoblasts? So osteoblasts are stem cells that they are fused in order to form each muscle fiber early in the development, and each contributing one nucleus. Each is contributing one nucleus. There are satellite cells. These satellite cells, these are unspecialized myoblasts that are remaining between the muscle fiber and the endomysium. So they play a role in the regeneration of damaged skeletal muscle tissue. They play a role in the regeneration of the damaged skeletal muscle tissue. So myoblasts are stem cells that fuse to form each muscle fiber early in the development, and each one is contributing to the nucleus. Guys, you have to know your myoblasts. Your myoblasts are your muscle cells that will divide. These are the stem cells that are fused to form the muscle fiber early in the development. What are the satellite cells? Satellite cells are out in space. Satellite cells are type of myoblasts that remain between the muscle fiber and the cover. You see the cover, the sheath that's outside the muscle fiber, that is your satellite cells. They play a role in the regeneration of the damaged skeletal muscle tissue. They play a role in the regeneration of the damaged skeletal muscle tissue. Okay, the structure of a skeletal muscle fiber. This is the structure of your skeletal muscle fiber. There are myofibrils, and the myofibrils have a lot of myofilaments. So you see these small ones coming out, these are your myofilaments. And these big regions are your myofilaments, while these are your myofilaments, and these are your myoblasts. The big ones are the myoblasts. So the myoblast has multiple myofilament. See, that's the myoblast, and the little ones are your myofilaments. Then you have your bands. You guys see your bands? That's your I band. We're going to talk about that. The I band is the light band, and the A band is, so actin is thin. So you have the light band is the I band, and the thick band is the A band. We will see more about that. Okay, now let's talk about these myofilaments. So what's bigger, the myofibrils or the myofilaments? So the myofibrils are big because they contain multiple myofilaments. So myofibrils are composed of muscle filaments or myofilaments, and there are three kinds of myofilaments. We have three kinds of myofilaments. So these are your myofilaments. There's three kinds. There's thick myofilament, and the first type is the thick myofilament because it is made up of several hundred myosin molecules. Several hundred myosin molecules. So myosin is a motor protein. So each molecule is shaped like a golf club that contains two chains that are intertwined to form a shaft-like tail and a double globular head. So the heads are directed outward in a helical array around the bundle, and the heads on one half of the thick filament angle to the left, and while heads on the other half angle to the right. And the bare zone has no heads in the middle. So if we examine here, what is that? Do you see the golf heads? These are your golf heads. And this is the tail. You see the long tail, and these are the heads. This is myosin. So myosin has two golf heads and two tails that are intertwined. This is really important. And then multiple myosin will give you a thick filament. Multiple myosin will give you a thick filament. Multiple myosin will give you a thick filament. So thick filaments are, so there we go. These are our myofilaments. And the first type of the myofilaments is the thick filament. So thick filament has two heads that are attached to each other with two intertwined tails. Do you guys see the two intertwined tails? And we have a thick filament because the thick filament is made up of multiple myosin molecules. Does that make sense? You have to recognize this is myosin. It has two golf heads and two tails that are intertwined like braid. And then the more myosin you have, the more darker, the more thicker you will have, the more thicker the myofilament will be. So when you say there's a thick filament, you know because it has a lot of myosin. Now, when we look at thin filaments, do thin filaments have myosin? No, they have actin. So thin actin, thin actin. So it has IN, thin IN, actin IN. So the thin filaments are composed of three different protein types. You have fibrous F actin, fibrous F actin. You have tropomyosin, and you have troponin. That is very important. The actin is the actual contractile protein. So it is actin who will bind myosin. Actin myosin will bind. These are known as contractile proteins. But tropomyosin and troponin, they are not contractile. They are regulatory. They are accessory. They help actin and myosin to contract. So the accessory proteins, the regulatory proteins, they are helping the contraction take place, but themselves, they are not contractile. So you have the fibrous F actin. It is composed of two intertwined strands of globular actin. That's why we call it G actin, because it is made up of a protein known as G actin. Globular protein, giving us G-actin, each with an active site. So it has a hole, an active site that the Golgi head will bind to. Remember we said myosin has a Golgi head, so the Golgi head will bind the actin, and that's how they're going to contract. So you have, and then you have tropomyosin that will block 6 or 7 active sites on the G-actin, and troponin is a small calcium binding protein on each tropomyosin. So on the exam, the question is going to be, which of these proteins is contractile protein? You say my Golgi head myosin and the actin, the red G actin. Who are regulatory, but they are not contractile? You say the regulatory proteins that are not contractile, but they regulate the contraction, they help the contraction. These are tropomyosin and troponin. Both of them have tropomyosin and troponin. So each would block 6 or 7 active sites on the G-actin subunit. So troponin is a small calcium binding protein on each tropomyosin molecule. Okay, the thin filament. The thin filament is composed, what do we say the thin filament is composed of? Does it have myosin, Golgi heads? There is no myosin in the thin filament. The myosin is only found in the thick filament. So now let's examine our thin filament. That is really important. The thin filament has the red balls, these red tomatoes. What are these? These are your G-actin. These are your G-actin. These red balls have holes inside them. They have holes known as the active site. But these holes are covered. They are covered by who? Who's covering the holes? Do you see holes now on the red? Do you see the red balls, the red actin? Do you see holes? Is not, because who's covering them? The troponin complex. So the troponin complex is covering the hole. But when calcium binds the troponin complex, remember, you see, troponin is a small calcium binding protein. So on the exam, who's the protein that binds calcium? It is the troponin. So when calcium binds troponin, troponin is going to drag the rope away, is going to move away. And then who can you see now? The holes, the active site. Who is going to bind the active site is your Golgi head, the myosin. So now you can never have a contraction because tropomyosin rope and the troponin complex are covering the hole. But when calcium binds troponin, troponin is going to drag away, exposing the site. So now let's look at that. Now there we are. Who's the purple? Do you see your golf heads? Do you see your golf heads here? Do you see these golf heads? You see these golf heads? These are your what? Myosin. So the purple is what? Thick filament. That's a thick filament. Is the purple golf head myosin so why is it thick? Because it has so much myosin. But what about the red? It is so thin because it only has the red balls actin G actin, and it has tropomyosin and it has troponin. So in the middle zone is called the Bayer zone. Why did they call it the Bayer zone? Because it has no actin and it has no myosin. It has no actin, it has no myosin. So there's an overlap of the thick and the thin filament. There's an overlap of thick and thin filaments. So there are three kinds of myofilaments. There is an elastic filament. There's an elastic filament that is made of huge, springy protein called titin. Is a huge springy protein called titin. So titin is going to run through the core of the thin filament and anchor it to the Z disc and the M line. And that would help stabilize and position the thick filament. And that's going to prevent the overstretching and provide the recoil. So of the myofilaments, myosin and actin are the contractile proteins. Myosin and actin are the contractile proteins. They do the work of contraction. They do the work of the contraction. So we said that. Remember, we said that the myosin and the actin are the contractile proteins. Myosin and actin are the contractile proteins, and they do the work of contraction. While tropomyosin and troponin act as regulatory proteins. Tropomyosin and troponin would act as regulatory proteins. So they act like a switch that determines when the fiber can and cannot contract. They determine when can the fibers act and they cannot contract. So the tropomyosin and troponin act as regulatory proteins. We talked about that. We talked about the fact that they are regulatory proteins. So the contraction activated by the release of calcium into the sarcoplasm and its binding to troponin. Then troponin changes its shape and moves tropomyosin off of the active site on the actin. There are several other proteins that are associated with the myofilament to anchor, align, and regulate them. So you have dystrophin. Dystrophin links actin that is in the outermost myofilament to membrane proteins that link to the endomysium. It transfers forces of muscle contraction to the connective tissue, ultimately leading to the tendon. There are genetic defects in dystrophin that produce disabling disease known as muscular dystrophy. So let's look at dystrophin. You see this red dystrophin. You see the red dystrophin. Dystrophin is binding the linker proteins. The linker proteins are on the sarcolemma. They are on the sarcolemma, on the plasma membrane, and they are binding to the actin. They are bound to actin. So you see, red is actin. So actin is attached to the linker proteins on the plasma membrane with dystrophin. With dystrophin. There are striations and sarcomeres. In cardiac and skeletal muscle cells, myosin and actin are organized in precise array that accounts for striations. So striations are alternating A bands that are dark and I bands that are light. A bands that are dark and I bands that are light. So the A bands are dark, and the darkest part is where the thick filaments would overlap, a hexagonal array of thin filaments. You have H band and the M line. We will see this more in the figure. So the I band is a light band where you would have a Z disc, and the Z disc, from each Z disc to the next Z disc, is the sarcomere. So let's look at this structure here. This is a very important structure. Now do you see why we call the A band the thick band? The A band is the thick band because it has a lot of thick filament. You see, you have a lot of thick filament. But if you look in the middle here, there is no myosin. There is no thick filament. There's no thick filament. There's no myosin. There's no thick filament. So what does that tell you? That tells you that this really thin line that is in the middle, that is the bare zone. So the H band is the bare zone. It has no thickness because it has no myosin. Then if you look outside in the I band, in the I band, in the I band, you have your, in the I band, you have, these are the I bands. I bands do not also have any form of myosin. But your sarcomere is going to extend from this Z disc that is found within the I band. So if you look at the I band, there's a Z disc here. If you look at the I band, there's a Z disc in the middle. And if you look at both of the Z disc here and the Z disc here, and you would see that this is a very light, very thin band known as the I band. But from each Z disc to the next Z disc, from each Z disc to the next Z disc, what do you have? What does that give you? From one Z disc to the next Z disc is your sarcomere, and that is your contractile unit. So from the Z band, from the Z disc to the next Z disc is your sarcomere, and when the sarcomeres contract, that is going to cause the contraction of the muscle because the sarcomere got smaller, sarcomere got shorter. Okay, so the sarcomere is a segment from the Z disc to the Z disc. We agreed to that. We have discussed that the sarcomere is a segment from the Z disc into the next Z disc. Sorry. Okay.
Yeah, you're gonna get a break at 5. I'm sorry, at 4:45.
Okay. So from Z disc to Z disc, what do we have? That is our sarcomere. So the muscle cells shorten because their individual sarcomeres get shorter. They shorten. And the Z disc or the Z lines are pulled closer together. They are pulled closer together as thick and thin filaments slide past each other. As thick and thin filaments slide past each other. So neither thick nor thin filaments can change the length during the shortening. Only the amount of overlap will change. So during the shortening, dystrophin and linking proteins also pull on extracellular proteins, and that transfers the pull to extracellular tissue. Now, when we look at the skeletal muscle cannot contract unless it is stimulated by a nerve. So if a nerve connection are severed or poisoned, a muscle is paralyzed. This is really important that you understand the concept of denervation atrophy. What is a denervation atrophy? Denervation atrophy is when you have atrophy because there is no innervation because of the denervation. What is atrophy? Atrophy is the opposite of hypertrophy. So when the athlete goes to the gym and they're working out, what is increasing inside their cells? The glycogen. Glycogen is increasing. That's why they say working out lowers blood glucose, because the sugar is going to go from your blood into the liver. The sugar is going to go from your blood into the muscle cell, There is denervation atrophy, and denervation atrophy represents shrinkage of paralyzed muscle when the nerve remains disconnected. When the nerve remains disconnected. So the skeletal muscle cannot contract. Skeletal muscle cannot contract unless stimulated by a nerve. So if the nerve connections are severed or poisoned, a muscle is paralyzed and may waste away, known as denervation atrophy. Known as denervation atrophy. So if the nerve connections are severed or poisoned, a muscle is paralyzed and may waste away. That is known as denervation atrophy. So when you get bitten by a cobra, God forbid, and the poison is destroying the muscle cells, what's going to happen is your nerve cannot innervate, cannot stimulate the muscle tissue, and that's why the muscle is going to get weaker. Nothing is stimulating me. So that's why people who are suffering from lower limb paralysis, you see that their legs are really small and tiny compared to their arms. Why? Because the arms are innervated properly, no problem. It is the legs that become really, really thin, because it's the concept of if you don't use it, you lose it. So same thing, if you don't use your muscle because it's not being stimulated properly with the nerve, then that's going to cause loss. So you don't use it, you're going to lose it. And the nerve cells whose cell bodies are in the brainstem and the spinal cord that serve the skeletal muscles. So their axons are called somatic motor fibers that lead to the skeletal muscle. And each nerve fiber branches out to a number of muscle fibers. So each muscle fiber is supplied by only one motor neuron. So the motor unit is when one nerve fiber and all the muscle fibers innervated by it, they behave as a functional unit. So here, the same way you have a motor unit is like right now, you have one professor teaching a number of students. That's a learning unit. In the body, you have one nerve innervating multiple muscles. One nerve innervating multiple muscles. So that problem now is going to show that in your skeletal muscles, if you have one nerve innervating 3 or 4 muscles, while one nerve innervating 1,000 muscles, how is that going to affect the specificity? When you close your eyes and touch with your fingertips, you're going to feel a pin, a needle. But if you have that needle and you mash it a little bit on your back, you're not going to feel that tip. You're going to feel some vague feeling. You understand what I'm saying? So if you have a sharp object, you touch your index finger with it, you're going to feel it. It's very sharp pin. But if you touch it in the back, you're not going to feel it. You're going to feel just some vague, something that's really vague. Why is that? Because in the fingertip, you have one nerve innervating 3 to 4 fibers. But in the back, you have one nerve innervating 1,000 nerve fibers. So when you have one professor teaching 1,000 students versus one professor teaching 10 students, then what's the specificity level now? So the less students per professor, the more learning. So the same thing, the less muscles per nerve, the more fine the tactile reception will be. You understand? Is that concept clear? So the effective contraction usually requires the contraction of several motor units at once. So the average motor unit contains 200 muscle fibers distributed across as many as 100 fascicles. As many as 100 fascicles. So the motor units can vary in size. That is what we've been talking about. That is a small motor unit. A small motor unit provide a fine degree of control. You see, a small motor unit, like for example, in the eye and the hand muscles, these are controlled by motor units with only 3 to 5. So each nerve is innervating only 3 to 5 units. While in the larger motor units, we provide more strength. We provide more strength than control. So here you have, like in your quadriceps, you see the mouse? In your quadriceps and your gastrocnemius muscles, you have one unit that may innervate 1,000 muscle fibers. So here, that's the motor unit innervating multiple muscle fibers. One unit innervating multiple muscle fibers. So the neuromuscular junction. The neuromuscular junction, also known as the motor end plate. So the synapse is the point where a nerve fiber meets its target cell. So when the target cell is a muscle fiber, the synapse is also called a neuromuscular junction or a motor end plate, a motor end plate. So the synapse is a point where a nerve fiber would meet its target cell, and when the target cell is a muscle fiber, the synapse is also called a neuromuscular junction. So we're going to see this in a little bit. When a nerve has a synapse with a nerve, that is not a neuromuscular junction. It is when the nerve has a synapse with a muscle, that is your neuromuscular junction. So each terminal branch of the nerve fiber within the neuromuscular junction would form a separate synapse with the muscle fiber consisting of terminal axon terminal, that is a swollen end of the nerve fiber, and it contains synaptic vesicles. Synaptic vesicles meaning that they can go in the synapse. That's what they call them, synaptic vesicles, with neurotransmitter called acetylcholine. So inside this synaptic vesicle, you have acetylcholine. You have acetylcholine. So the nerve impulse causes synaptic vesicles to undergo exocytosis, releasing acetylcholine into the synaptic cleft. So the nerve impulse would cause the synaptic vesicles to undergo exocytosis, releasing acetylcholine in the synaptic cleft. So the synaptic cleft is a narrow gap. It is a narrow gap between the axon terminal and the sarcolemma. It is a narrow gap between the axon terminal and the sarcolemma. That's the synaptic cleft. We're going to see it. There we go. Guys, you really need to pay attention now. This is so important. If you cannot see far, please look at that TV. So what's going to happen here? What do you see in A? What does that look like? We talked about that. What is this where my mouse is? Do you see the mouse? Where my mouse is moving. What is that? I'm sorry? That's a muscle. What is the purple circle? I asked that in the beginning. When I asked you what is that, and nobody answered the question. When we were this is what I'm asking now. So what is that? What's the whole thing? That's a muscle fiber. So when I look here, I see my muscle fiber. That is my muscle fiber. You see, this is my muscle fiber. Now, what is this yellow? These are my swollen ends. You see, the axon terminal, swollen end of the nerve fiber. You see the swollen end? That is the swollen end. What is the swollen end touching? The muscle. This is right here. So now we showed you, that's my swollen end, which is part of my nerve. That's part of my nerve. And what is that? This is your, what is that? This is your, what is this side, guys? This side is the nerve. That's the swollen end of the nerve. Below it is your muscle. So that's the nerve. What's below it? Here, this whole region, all the region. Yes, receptors are right here, but this whole compartment, if this whole up top is your neuron, then down here, what is that? The synaptic cleft is in the middle. That's the gap. I'm not pointing to the gap. I'm pointing here, this whole region down here. That's the muscle. You see, that's the nerve and that's the muscle. Then between them is the gap known as the synaptic. That's the synaptic vesicle. So that means I need to empty. I need to secrete. I need to send out. Empty, secrete, send out my acetylcholine. So the acetylcholine is going to be sent out. You see, the acetylcholine is going to be sent out, and the acetylcholine is going to bind the acetylcholine the acetylcholine receptors. So where do we find the receptors? We find them in the muscle, in the postsynapse. So that's my nerve, What are these guys? The yellow ones. What are those? These are your swollen ends of the nerve. And what is that here? And what's the purple? Nucleus. Then when I zoom in, you see that's why they made the box here. They put the box for you to show you that we are zooming in. We are zooming in. When we zoom in, we have when we zoom in, we have this gap, and this gap, you have your receptors, and the receptors would bind the acetylcholine that is going to be excreted out. Acetylcholine that is going to be sent out. Okay. Structure and function of the neuromuscular junction. What is the structure and the function of the neuromuscular junction? So the muscle cell sarcolemma has millions of acetylcholine receptors. Right here in the sarcolemma, you have millions of receptors. When you have no receptors, what's the name of that disease? The disease where you have no acetylcholine receptors. I'm sorry? I'm sorry? Alzheimer? Alzheimer is when the nerve is damaged. The nerve is stressed out and injured. But here, we're not talking about the nerve. We're talking about the receptor on the muscle. The receptor on the sarcolemma. If this receptor is no longer there, what is that called? Yes. It's called myasthenia gravis. Myasthenia gravis or myasthenia gravis. Okay. So now let's look at the postsynaptic junction. You have invaginations. What's the purpose of these invaginations? Foldings. That reminds you of the intestine. Remember, the intestine has many microvilli that folded. That is to cause what? That is to increase the surface area. So we have invaginations to increase the, there you go, that junctional fold. There are postsynaptic membrane fold of the sarcolemma that increase the surface area, that maximize the number of acetylcholine receptors. The lack of receptors causes weakness in the disease known as myasthenia gravis or myasthenia gravis. So the entire neuromuscular junction is enclosed in the basal lamina. It is enclosed in the basal lamina. It is a thin layer of collagen and glycoprotein, and separating the muscle fiber and the nerve ending from surrounding the connective tissue. So it passes through the synaptic cleft and nearly fills it. So the sarcolemma and the lamina with the synaptic cleft contain acetylcholinesterase. Acetylcholinesterase. So the enzyme that breaks down acetylcholine, allowing for the relaxation. It's an enzyme that breaks down acetylcholine, allowing for the relaxation. So this acetylcholinesterase, it ends with an ACE, cholinesterase. So what does ACE tell you? It's an enzyme. So it's an acetylcholinesterase. That means what does it do to acetylcholine? It's gonna break it down. So you know, in South America, the native Indians, they have the blow dart. They blow on it, and then it hits you in the leg. You cannot move no more. They take you, they eat you up. You've never heard of that? You never heard of the South Indians blowing the thing? It hits you in your foot. It paralyzes you. You can't move. They catch you. They take you. They eat you. Okay. So how did they do that? Because the liquid they have in the thing they blow is called curare. it will competitively compete with acetylcholine. It will compete with acetylcholine, and when it binds the receptor, it will not do what the acetylcholine do. What does acetylcholine do when it binds the receptor? It's going to open the channels to cause calcium to go and bind troponin and cause the contraction between actin and myosin. But curare from the thing that they blow is going to bind. It's going to bind to the receptor, but it's not going to cause calcium release. So you're not going to have contraction. You cannot move. So that's a natural, something natural that they find. They get it from a poison. They get it from the poison of the plants. So the issue here is that you're supposed to have acetylcholine emanating, coming out of these vesicles, binding these blue receptors, and when they bind the blue receptors, the calcium is going to bind. Remember the red actin is going to cause the contraction. Then you contract. You know, when you move, you contract your muscles. But when they blow that thing and then it hits you, what's going to happen is, instead of the green acetylcholine, you're going to have something called curare. Curare. Curare is going to bind the blue. Blue doesn't know who's going to bind it. If N-acetylcholine binds it, it's going to do that. If curare binds, it's going to bind, but it's not going to be no calcium because it's not acetylcholine, it's curare. And that's going to keep you in place. Yeah, it doesn't allow for this whole cascade. Because remember, we need acetylcholine to bind the receptor. Then after binding the receptor, you're going to have secretion of calcium. Then calcium is going to leave the cistern. Remember the cistern in your cell? And then they're going to bind to the troponin complex, removing tropomyosin, exposing the gold holes so that the gold heads, they're going to bind to the actin and the contraction happen. You can move. But with curare, that cannot happen because it doesn't cause calcium. Yeah. Yes. Similar. Okay. Yes. Yeah, it wears off. But by the time it wears off, they already caught you because they're right around the corner. Okay. They're looking. Oh, Jennifer. And by the time Jennifer says, okay, no more, okay, they're going to wear off, and then I'm going to be able to walk again, they caught you. Yeah, that's what they do. They are awful people. Yeah, in the South America, in the group, in the tribes, there's some tribes, they still do it. They don't care, they still do it. And it's now, it's country. They have multiple countries. And the government know about them, but they can't control them. Just like here, you have gang violence, you can't control that. You can't control the problem gang violence. They just leave that out. Because if the police go there, they're gonna eat the police. They're gonna eat the police is not going. So that's very, very, they're eating people up to now. They used to do this thousands of years ago, they still doing it. And the thing is, human food is dangerous. If you eat a human being, it's gonna kill you. The lion can eat the human being because he's got enzymes that can do that. But your body is only used to eating soft things, like sheep food or like something that. But you can't eat human being. You want to eat food. Even if they cook it, it's still bad. Because the human being, when you is so stressed out, it knows you hit me with the thing, you're gonna eat me. I'm so stressed out, and the stress enzymes and the stress receptors are secreted all in my body. That's why when they kill the cow by smashing the head and doing all that kind of stuff, the cow is so stressed out. Now its meat is so toxic, so you eat it, you die. Be careful. Yes. So I don't think it's healthy for them, but you need to adapt. Yeah.
body's adapted to it. But I don't think their lifespan is that long. They don't last a long time going around. They die maybe like very young age. But that's a terrible kind of thing, you know, to eat a human being. Yeah, very bad. Hmm? It's bad to them. Yeah, it's toxic. Human food is toxic to humans. It's not toxic to animals. We don't have the enzymes so a cow cannot eat a cow. The lion cannot eat the lion. But the lion can eat the sheep and a human being. We only eat down the food chain. We don't eat up the food chain. We're not gonna go eat up a lion. That's gonna hurt you. Cats and dogs. Yes. They are programmed, yeah, they will not do it. They will kill a lion, but they will not eat it. It is the hyena who will eat it. Because that's scavengers. Vulture, yeah, vulture. Yeah, I hate vulture. Vultures are bad. So yeah, this is very dangerous. It's called cannibalism. I do not want. That's why I've never gone there because I don't want them to eat it. Here they cannot. They cannot go on the subway. It's gonna be fine here. But if you go there, be careful or don't go to the jungle. Actually, some people did a documentary. They went, they sat with them and talked to them. Yeah, if you Google it, how did we know about all of this? It's because of the people who go to them. Yeah. And there is tribes that drink blood that's in Kenya in Africa. It's called the Maasai. They drink the blood. I think they drink, yeah, human, cow, all that, yeah. It's very bad. Okay. So guys, this is really important. It's really important that you see this. This is a process that you really got to walk out with today. That the first step is the action potential is going to come. Please look here. This is really important. The action potential is going to come in here, and the action potential is going to cause the calcium is going to enter the axon. That's in the nerve. So the action potential is going to make calcium enter inside the swollen end of the nerve. This calcium is going to cause, so first of all, you have an arrival. What's the first step? Where my mouse is? This is the arrival of the nerve signal. Then you have acetylcholine that is released from the synaptic vesicles. Acetylcholine is now going to go and is going to bind acetylcholine receptors, is going to bind the acetylcholine receptors, and these acetylcholine receptors are going to open and the acetylcholine would be released, and the ions would flow inwardly, and the binding of the calcium to the receptor. So the binding of the calcium to the receptor would cause the opening of the ligand regulated ion gates, and there's a creation of end plate potential. You see, there's a creation of end plate potential when sodium influx and potassium would efflux. And then the opening of the voltage regulated ion gates. That would cause the creation of action potential. You see, sodium is entering into the cell and potassium is leaving. So you see sodium entering, that's depolarization. Potassium exiting, that's repolarization. So the opening of the voltage regulated ion gates would create action potential. So you see, opening of ligand regulated ion gate creating a end plate potential. So you see, that's the plasma membrane of the axon terminal. You see, the plasma membrane, you have acetylcholine binding to the receptor, leading to the opening of the sodium and potassium channels. Sodium would enter inside and potassium would exit. Yes.
When it binds, you have one side has the sodium and the other side has the potassium.
Yes, so when can you please look there? When acetylcholine binds to the acetylcholine receptor, what happens is you have opening. You have opening of these channels, and the opening of these channels leads to the entry of sodium, and the exit of potassium. Entry of sodium and exit of potassium. And that is, these are known as the voltage regulated ion gates. And then there's the opening of the voltage regulated ion gates would cause creation of action potentials. Would cause a creation of action potentials.
One of them was
Yes.
it's like when it opens, it has that little one. It's like two channels. One goes in, one goes out.
Yeah. No, but they are in order. The first one that opens, I know it's not shown here. This is not a clear diagram, but this here shows you that sodium channel would open first, causing the influx of sodium, and then the potassium channel would open, causing the efflux of potassium. Clear?
Yeah.
Then you have the action potential propagated down the T tubule. So you see, when you cause the binding, I don't know why you guys are not following. What's happening here? When the vesicle buds off the terminal and acetylcholine would go and bind to the acetylcholine receptor, that's going to propagate an action potential. The action potential is now going to go and is going to bind to the T tubule. Is going to bind to the T tubule. And the action potential propagated is going to be propagated down the T tubules. So calcium is released from the terminal cisterns. So calcium is released from the terminal cisterns. And the terminal cisterns are going to cause calcium secretion, and calcium is going to bind troponin. And there you go. You see what happens when calcium binds? Are you guys following here? When calcium binds, what happens? You see the arrow moving up or down, exposing. With no calcium, there is no holes. With calcium, everything moved away, and now you can see the holes. Now you can see the holes. And then in these holes, who's going to bind? Myosin. And that's going to cause the shifting of the tropomyosin, exposing the active sites on the active. Exposing the active site on the active. So here we see that hydrolysis of ATP to ADP is the activation, initiates the activation and the caulking of the myosin heads. Hydrolysis of ATP to ADP causes the activation and the caulking of the myosin heads. So the formation of myosin actin cross bridge. Formation of the myosin actin cross bridge. So here we made a cross bridge between the myosin and the actin. There is a cross bridge between the myosin and the actin. And then that would cause a power stroke. You see, the power stroke is when we are sliding the thin filament over the thick filament. Is when you slide the thin filament over the thick filament. Sliding of the thin filament over the thick filament. Then there is binding of the new ATP would break the cross bridge. So that's our cycle. You see, it is the ATP that causes the relaxation. So ATP causes the relaxation and the breaking of the cross bridge. But ADP causes the formation of the cross bridge and causes the formation of the cross bridge and causes that movement, that locking movement. So hydrolysis of ATP to ADP causes the activation and cocking of the myosin head. So the formation of the myosin-actin cross bridge leads to the power stroke, sliding of the thin filament over the thick filament, causing the binding of the new ATP and the breaking of the cross bridge. Cessation of the nervous stimulation and ACH release. That causes the cessation of the nervous stimulation and ACH release. And here you have ACH breakdown by acetylcholinesterase. This is the reabsorption of calcium ions by the sarcoplasmic reticulum, and there is a loss of calcium ions from troponin. You see, calcium would start to remove in order for us. And now you see the arrow is going to now return back. So there is a return of the tropomyosin to the position blocking the active sites of the actin. This is very important. So I'm going to stop here. You guys can go have your break.